Today, we are going to discuss about molecular basis of infection and disease resistance or defense. Being sessile organisms, plants are often exploited as a source of food and shelter by a wide range of parasites, including viruses, bacteria, fungi, nematodes, insects, and even other plants. However, plants have developed remarkable strategies to adapt to environmental changes by using a range of constitutive or inducible biochemical and molecular mechanisms. Plants exhibit both long and short term defense responses to immediate challenges such as pathogen attacks. Once pathogens penetrate the cell wall, the plant's two-tied innate immune system is activated to counter attack pathogen invasion. Whether the pathogen can infect a plant or not depends on the genes for pathogenicity and virulence present in the pathogen and the genes for resistance or susceptibility present in the plants. In plants, the estimated loss caused by pathogen is typically around 10 to 20%. Molecular basis of infection and disease resistance. Plant disease resistance is generally divided into two types: qualitative and quantitative resistance. Qualitative resistance is controlled by major R genes, and the quantitative is conditioned by multiple genes with minor effects. R genes usually confers complete resistance to a specific pathogen or pathogen race and is easy to manipulate for basic research and crop improvement. As the resistance mediated by major R genes can be rapidly overcome by new virulent pathogens, so R gene represent a frustrating battle in disease control for plant breeders and farmers. Quantitative disease resistance is controlled by multiple genes, each contributing to partial resistance. Quantitative disease resistance leads to lower selection pressure against pathogen variants, and those that overcome an individual quantitative resistance locus have little advantage. Thus, quantitative disease resistance tends to be more durable than R gene mediated resistance. Next is major genes for disease resistance. At the molecular level, defense system often depends on the combination of a specific set of dominant R genes in the plant and a corresponding set of dominant A virulence genes in the pathogen. Expression of A virulence genes triggers plant defense responses governed by the product of the R gene. This gene for gene resistance strategy underlies the molecular basis of defense system in plants. When the plant is resistant and the pathogen is said to be avirulent and the interaction is said to be incompatible. When the plant is susceptible, the pathogen is said to be virulent and the interaction is said to be compatible. In a given plant pathogen interaction, it is often possible that more than one specific combination of A virulence and R genes are operating at the same time, and such different combinations are often codominant. These multiple combinations reflect the complexity of defense mechanisms of the plant during pathogen attack. R genes can be generally divided into 7 classes based on their amino acid motif organization and membrane spanning domains. The largest class consists of NBS-LRR genes, which contains a nucleotide binding site, that is NBS, and leucine-rich repeat LRR domain. NBS-LRR genes in plants are typically divided into two groups according to their N-terminal domains. One is the TIR group genes that are composed of N-terminal domain, and other group of genes code for CC, that is coiled domain. So there are about 200 genes that encode proteins with the similarity to nucleotide binding site and other domains characteristic of plant-resistant proteins in Arabidopsis. Second class is characterized by extracytoplasmic LRRs and C-terminal membrane anchor represented by the tomato CF genes. The rice XA21 gene, which confers resistance to a bacterial pathogen, is characteristic for the third class of R genes. as it features both extracellular LRRs and a transmembrane protein kinase. The Arabidopsis RPW8 protein is an example of the fourth class of resistant gene encoding proteins, which contains a membrane protein domain and a putative coiled coil domain. The fifth class of resistant gene encoding protein contains putative extracellular LRRs, a PEST domain. The sixth class is represented by the RRS1R gene in Arabidopsis, which has C-terminal extension with the putative nuclear localization signal NLS and WRKY domain. The seventh class encodes enzymatic protein, which contains neither LRR nor NBS domain. Next is mechanisms underlying plant resistant to pathogens. Plant pathogens are broadly divided into two types, biotrophs and necrotrophs, according to their lifestyles. Biotrophic pathogens gain nutrients from living host tissues, whereas necrotrophic pathogens kill host tissue and feed on the remains. The molecular mechanisms underlying activation of plant defense responses are quite different between biotrophs and necrotrophs. Plant defense against biotrophic pathogen is largely due to gene-to-gene resistance. Our gene-mediated resistance usually result in hypersensitive response, which is thought to be very important for plants to combat biotrophic pathogens. Our gene-mediated resistance also activate assay-dependent signaling, leading to activation of string of presumed defense effector genes. Generally, it was assumed that no gene for gene resistance function in resistant to necrotrophic pathogen, but it was indicated in a number of reports that several plant pattern recognition receptors, PRRs, are involved in the perception of necrotrophic pathogen like fungi. So just look at this diagram. Here you can see here RLKs. These are receptor-like protein kinase present on the surface of the cell, and they recognize the necrotrophs. Then HSTs, host selective toxins, they are considered as efficient weaponry of necrotrophic pathogen like fungi. In plants, then a group of small cysteine-rich protein called defensins, here this, it display antimicrobial activities, and these defensin are encoded by pathogens related genes, that is PTF 1.2 genes here. So function of this defensin is to reduce hyphal elongation and triggering fungal membrane permeability, and these all leads to suppression of necrotrophic fungi growth. So these a pathogenesis related gene, this PDF 1.2 gene encoding this defensin in Arabidopsis was regulated by zasmonic acid, JA. So JA and ethylene mediated defense responses are expected to play a key role in resistance to necrotrophic pathogen. So all these lead to immune responses. Second is a two-tiered innate immune system in plants. So as I already told you, plants lack mobile defender cells and a somatic adaptive immune system. Instead, they have evolved an innate immune system. So there are two tiers in plant immune system. The first tier is based on the sensitive perception of PAMPs, pathogen associated molecular patterns. Through PRRs, these are receptors, means pattern recognition receptors on the cell surface of the plant. So immune responses to PAMPs are categorized as first tier of defense. Now, successful pathogens have evolved to produce effector to inhibit first tier of defense, and the plant can perceive such effectors through additional receptors, R protein, to form the second tier of defense. So there is a dynamic co-evolution between plants and pathogens, and this dynamic process continues as some pathogens have acquired effectors that interfere with the second tier of defense. So here, in the case of first tier of defense, as the first tier of plant resistance system is triggered by PAMPs. The leucine-rich repeat receptor-like protein kinases RLK named FLS2 binds to bacterial flagellin. FLS2 initiate immune signaling by association with another leucine-rich repeat receptor like kinase BAK1. So a receptor-like cytoplasmic kinase BIK1 is rapidly then phosphorylated upon flagellin perception by both FLS2 and BAK1. Phosphorylated BIK1 then transphosphorylates FLS2 or BAK1 to propagate flagellin signaling. A complete MAPK cascade and downstream transcription factors are then activated after FLG22 detection, which activates the defense response. So here look at this diagram. Here, in the case of first tier of defense, it is triggered on perception. Just look at this. These are the biotrophs. And this is triggered on perception of PDAMPs. That is, pathogen damage associated molecular patterns. And this bimembrane anchored PRRs, that is pathogen recognition receptors present here on the surface of the cell. And this is followed by activation of MAPK cascade and downstream transcription factors then leading to the immune responses. Now, in the case of second tier of defense, which is elicited, second tier of defense is elicited by pathogen effector via an interaction with R proteins. So here, so in second tier of defense, just look at this diagram. Here, plants evolved R genes to detect pathogen effectors and trigger defense responses, and pathogen in turn have evolved new effectors to evade second tier of defense. Now, such interactions between R proteins and these are R proteins and these are effectors. So, interaction between R proteins and effectors oscillate between compatible and incompatible reactions over time. So, genes encoding pathogen effectors that induce R gene mediated resistance are defined as virulence genes, which qualitatively reduce virulence, but only when the host has the cognate R gene. So, R genes, these R genes, these are mediated R gene mediated resistance to biotrophic pathogen usually result in hypersensitive response and meanwhile activates assay dependent signaling leading to systemic acquired resistance, that is SAR. Thanks for watching. If you like this video, please make sure to like and subscribe for more. Thank you.